the whole idea of type 1 diabetes, it's a very complicated challenge. So there's not sort of one magic bullet, but really as sort of the engineer in me, and it's part of what we're doing, it's sort of trying to assemble the right pieces. The first part of this is the microporous scaffold that sort of creates a home for the islets and the islet organoids that we're working with. The second component actually is working with the stem cells, to use the stem cells to actually produce the islets. The third component are these nanoparticles that you can use to sort of modulate the immune response, to essentially prevent the cells from being rejected. And then the fourth component are these sort of non-invasive imaging technologies that are really about trying to identify problems before they happen with the cells that you've transplanted. Why don't we get started? If everyone, and I have 4 stations in the lab, and there are a bunch of graduate students and postdocs that are back here that I think we'll try to split you up into 4 groups. Thank you all for coming. We spend so much time thinking about this. It is really great to see people that really care about what we're doing as well too. We've been working in sort of regenerative medicine right from the start. So I had done my PhD and a postdoc here, sort of in the idea of biomaterials for tissue engineering. And the idea is the biomaterials are sort of these platform tools that you tune to really control cell-cell interactions. You can do drug delivery. We've done gene delivery. We've done some nanoparticles, so I'll sort of touch on these things. And that's something that we've been able to take to sort of a number of different applications. And I think the major idea is like this is a unique set of tools to what you will see that are available if you talk to just the biologists or you talk to the clinicians. We're very complementary in terms of providing tools that most people do not have at their disposal. So in addition, we love alliteration, so we have different subgroups, and so the glucose gang is who you'll be hanging out with today in terms of what they're doing. Our approaches are sort of illustrated here. They all start with this idea of a microporous scaffold for delivering the cells, but these are the stations that I'm going to have you go through. So the microporous scaffold, it's really the idea of really trying to create a home for the cells that you transplant. The cells are obviously vitally important. They're doing all the function that you need them to do, but if you put the cells into a bad environment, they might lose their ability to function. And so the scaffold is really sort of the right support for them to be able to do the functions that they need. Don't be nervous.
I usually wear glasses, so that's why probably.
You have a friendly audience.
Right. Okay.
No blame.
Well, except for Elizabeth, she'll grill you.
How big is it going to be when it's done?
How big?
Yeah.
He'll show you the process, but he's got some at the end that you can just pick up and see.
I will have sample too.
Pass them
Yeah,
around.
pass them around.
So this is a completed one.
Wow. That's tiny.
Well, so that's the mouse-sized one.
Oh, how big would a human one be?
So the human one is going to be more about
Wow.
25 to 35 millimeters in diameter.
Okay.
And so
So about 3 centimeters.
3 centimeters.
About
Exactly.
an inch.
Yep.
For those of us who do imperials.
So I wasn't doing the transition very well in my head, but yeah, absolutely. And so the idea is that they would go into the omentum, and it's just the idea that you have lots of space there, and it also has some natural wound healing properties to it. And so the idea of it goes in a laparoscopic procedure, so therefore it's not terribly invasive either.
Great.
So we put them under pressure, 5K pounds for 30 seconds to press them together. So as I said, they're the mix of PLG particles and table stock. Right.
I remember from high school chemistry, college chemistry, not so much.
Now we're getting them out of the dye.
Yeah, it's a process that they go through to make each one.
Yeah, it takes, they're time intensive, but they work very well, so they're worth it.
30 seconds doesn't seem too bad.
No.
And there are ways of scaling this, particularly if you make, if we weren't at a laboratory scale, you could imagine making a really large one, the sheet of it, and then cutting it into the size sheets that you would need. And so the scaling of it is easily done, but
Yep.
when you have
So
the
we're
resources to do that.
Right.
Finished scaffold. That's around.
The second part are the stem cell derived beta cells. And actually I would say stem cell derived islet organoids. And this is the idea that there aren't enough donor islets available to treat all the patients that are out there for type 1 diabetes. And so we need alternative sources of cells. And so we're working with culturing the stem cells to become these islet organoids. And we say organoids because they're sort of immature. They're not fully mature islets when we culture them in the lab. And so the organoids that we can grow in the lab, we can grow them on the scaffold that helps their maturation process in the lab, but then we can transplant them where they can actually complete the maturation process and turn from an islet organoid into a mature islet.
So we're going to go see the nanoparticles for treating autoimmune diseases. So I know that the focus here is largely diabetes, but we're also working on treatments for multiple sclerosis, looking at treating for transplants, and this is all important because all of these are major concerns, but also autoimmune diseases often have comorbidities. So it's often the case that a patient who has type 1 diabetes might have a comorbidity with another autoimmune disease, and so we want to be able to treat all of these. Okay, so now we're going to go into the particle fabrication lab.
This is the cell culture room. This is where I do most of my research, as Lonnie had talked about earlier. We're looking to see if we can use our biomaterials and stem cells in order to generate functional pancreas tissue. To kind of give you a quick general overview, the field of stem cell research is constantly growing, and I find super fascinating. My granddad had type 1 diabetes, so I kind of saw what it was like growing up as a patient. And as I got older, I wanted to contribute the best that I could, and as an engineer and scientist, investigate or interested in how the body develops. Stem cells and biomaterials was kind of like this really cool area that was developing regenerative medicine. So what I'm going to show you today is generally some 3D clusters. So these are pluripotent stem cells that have developed into 3D clusters that you guys will see, and then we incorporate those into the biomaterials to provide more supportive matrix. And so in general, the way stem cells have developed, first it was on a 2D culture plate, something that looks like a petri dish. I can kind of show you what that looks like. And that's been around for several decades. Essentially right here, this is an incubator that we have, keeps the cells at the right temperature, 37 degrees, 5% CO2. So you'd have a petri dish here is how we would generally culture stem cells. But generally what we have found is that we would rather culture them in a 3D model so it replicates more of how the body develops naturally. And so as a result, we've started to now culture them in 3D suspension in something that we call, one of the platforms that we can do 3D suspension in is called a spinner flask. And as you can see, it's appropriately titled because these propellers here essentially spin around, and as a result, clusters start to populate around. Now I'll get another spinner that's got a little bit prettier to look at. But in these spinner flasks, we can actually generate vast amounts of insulin-producing cells. So if you see these little dots and clusters floating around, you can see maybe up at the light, these clusters, these little dots, so those are clusters that we're now developing and differentiating into insulin-producing beta cells. And so as I said, you can have large vats of it. This is just 30 milliliters. Some people can have tanks as large as that tank right there, full of clusters. So it's a very scalable model that allows us to replicate more of that 3D morphology that the body naturally has. And then we take this one step further in our biomaterials by incorporating this 3D development within a biomaterial scaffold. And so these 3D clusters are no longer just floating in suspension, they are actually interacting with this matrix around to once again replicate how the body naturally develops. Obviously, we're not just a vast system of blood in between. We have tissue, extracellular matrix proteins that interact and develop and further help mature these cells. So our biomaterials are easy to tune, and as a result, we can actually further develop these insulin-producing cells.
When you put the clusters into the scaffold,
Yeah.
they're not fully differentiated at that
That's
point.
correct, yeah.
And that's why you create this
Exactly. So essentially,
cellular matrix that mimics.
Exactly. So essentially, they start out as a progenitor cell that can become pancreatic-like, and then when we put them in our biomaterials, we can actually further develop it. And to kind of show you, kind of give you more of a idea of what I mean by that, some context. As of right now, we can produce cells that are secreting insulin, but not in a glucose-responsive manner. So that means if you put these cells inside the body, the person can still deal with hyper or hypoglycemic conditions. What we're trying to do is develop cells that are actually glucose-responsive and therefore mature, and that way you can guarantee that they will reach that normal glycemia we're searching for.
How do you create an extracellular matrix that is like ours?
So it's a very complex way to create your own extracellular matrix. What's really nice about our model is we actually can take these progenitor cells, culture them within the scaffold, and they actually secrete their own matrix. They create their own complex environment, which is so much easier than us trying to go bottom-down approach and figure out, okay, how do we actually develop it? Because you're right, it is a very complex environment. And what's really interesting with our platform is we don't actually have to worry about those things. We can let the cells develop it as they naturally would within our inert material.
Does that enhance when you implant them?
It does, yeah. So as it continues to develop, it will continue to change that environment around it to help aid it in its function.
The third component were the nanoparticles that are really geared towards immunomodulation. And the reason why this is so important is that type 1 diabetes is an autoimmune disease, and so therefore your immune system gets dysregulated and ultimately destroys the beta cells. And then there's also the therapeutic side of it, is you're going to get cells to transplant. They're not coming from yourself, they're coming from someone else that might not have the same sort of proteins on them. That's not an exact match. So your body would tend to reject cells that are coming from another source. And so the idea of the nanoparticles is to be able to identify what are the proteins that are causing the autoimmune disease, or the proteins that are causing the rejection of the cells, that we are actually looking at ways delivering those proteins to get the body to accept those proteins and not respond to them, and therefore the cells could persist indefinitely in the body.
Within this vial we have an oil, a volatile oil. Within that oil is our antigen of choice. Now all I'm going to do is I'm going to add our water here. And what I want are little tiny droplets of the oil, because I want those little droplets to eventually become my PLG nanoparticles. And to get that, all I have to do, I'll pause for camera setup. All we have to do to get this is introduce a tremendous amount of energy locally to get little tiny droplets. So if you can see, it's becoming frothy white. It's just scattering light. So there's nothing chemically happening here. It's just all that oil is becoming tiny droplets, about 500 nanometers in diameter. And when we are done here,
Making it adhere together.
what's that?
Does it have to adhere together, the oil and the water?
No, in fact, they're just forming little tiny interfaces. So originally it's two layers. You got the oil and the water, and then we're just putting a ton of energy, shaking it really hard, like you would a salad dressing. You get little tiny droplets. And we actually want them to kind of stay apart so that we keep this nice 500 nanometer size particle there. And within that we have our antigen of choice. In this case, I work a lot on peanut allergy. That antigen was peanut. And so we will use these particles to then try to treat peanut allergy. But like Lonnie said, we've had a lot of success with type 1 diabetes models for treating the autoimmunity side. We're also working on ways, how do you overcome the transplantation? So if you gave me somebody else's islets, my body would reject them as foreign. So we can also use the same platform to deliver antigen from a donor to train them that, okay, this protein from this donor is also okay.
What's the commonality between peanut allergy, celiac, autoimmune type of?
The common thing that our platform really hits on is that all these things involve T cells that are attacking a protein that they shouldn't. And so with our platform, we can deliver the antigen to what we call antigen presenting cells. These are the cells that tell the T cell what they should or should not attack. So our particles talk to the APCs, antigen presenting cells. Those APCs tell the T cells, hey, this thing is innocuous, or at least they say this thing isn't dangerous. And then the T cells say, okay, I guess I won't attack this.
So that would that, like in the case of a peanut allergy, would that treat someone who's been exposed, or would it be given to prevent an allergy or a response from occurring?
Today is Wednesday. You'll have to ask me on Friday, because those studies are going on right now in mouse models. So we are actively engaging that. Prophylactically, the particles work for peanut allergy, for sure. We've shown that in mouse models, I should say. So now it's a matter of, all right, now obviously it's better if we can treat an existing allergy. So we do that.
And just for context, these particles have been licensed by a company, and they're in phase 2 clinical studies for celiac disease. And so once celiac, assuming we cure that, then it sort of opens the door to take it to all these other autoimmune diseases.
Is the idea that someone would have this treatment before their exposure or going forward, or
And so the idea is it prevention or is it therapeutic?
Yes, yes, yes.
And the idea is we've used them in both cases. And so it's a matter of knowing what the antigens are. That's, I think, is the hard part when you go to like a type 1 diabetes. And you can try to tolerize to the ones that you know are likely to develop, but then there might be other ones that you're not aware of. And so that's where the prevention, you can try to use the ones that you know of to prevent it. For the ones that you don't know what they are, that might be the therapeutic side that you need to put those in. The last demonstration.
There's no allergies. We just have a bunch of autoimmune.
In terms of type 1 diabetes therapies, you know, it is the most advanced cell-based therapy that's out there. And it, both the field needs it to happen, the FDA needs it to happen, industry needs it to happen. And so if you can if there's a success that can happen there, then it opens the door for lots of other things. And so I think type 1 diabetes, I think, is likely to be the first one. The challenge where I come from is being part of this encapsulation consortium. You see all these different approaches that are out there. And the encapsulation approaches, while I sort of said the pros and the cons of them, the pros and cons for us is we have to do something about the immune system as well. It's unclear which one of those is sort of going to win from a long-term strategy. But at the same time, I think that we've had so much success in those mouse models in terms of the efficiency of the cells transplanted and these other things in terms of like the fast ligand and everything, that there's, at the very least, maybe not the complete system ends up, but I think a portion of what we're doing will probably become part of what's ultimately going to go ahead, just because our technologies have been so successful.
Are you looking at years or decades?
Someone was asking me earlier, I mean, the nanoparticles obviously are already in phase 2 clinical trials. The scaffolds, we're collecting the data from the non-human primates to potentially start a clinical trial in the next 1.5 to 2 years. So I don't think it's that far off. The non-invasive imaging strategies are geared towards really having a system such that if you transplant the cells, you can monitor how they're doing without having to biopsy or isolate them. And the idea that you could essentially be able to image and identify that either the stem cells are developing appropriately or that the islets are fully functional. And that's where we had talked about some sort of the transcription factor reporters or being able to measure insulin release in the cells just by using a camera to detect the light emission using the imaging technologies that we've developed.
So we work with stem cells. So stem cells can turn into any cell type in the body, and we use very specific media formulations to direct the differentiation of these cells towards cell types that we would find in the islet of Langerhans. So as the cells progress, they go towards insulin-producing cells as well as other endocrine type cells. So one of the things that we want to do is to be able to understand what is the process by which cells are becoming beta cells and other cell types as well. So there's something in the cells called transcription factors. Transcription factors turn on and turn off genes, kind of like light switches. And we have a method in our lab where we can non-invasively, in real time, measure how these transcription factors are turning on or off certain genes. So my professor mentioned earlier firefly luciferase, which is how lightning bugs shine light. We use this same technology to study how cells are turning on transcription factors over time. So you can see here, these are 96 wells, and we can put cells into these wells with reporter constructs for different transcription factors. For example, we could look at beta cell transcription factors or alpha cell transcription factors and see at what time do these turn on. And this is really powerful because generally in lab techniques, cells need to be destroyed or frozen to be studied, and that only gives you a snapshot. Here we can understand what's going on throughout the entire process. So if I could direct your attention towards this screen over here, we have a reporter for pro-insulin. And this is really powerful because we can look into our clusters of cells within this tissue, and the high intensity yellow you see is stored insulin. It's insulin that's stored and ready for secretion. And we expose these tissues to high glucose, and over time, you can see from 1 minute to 2.5 to 20 minutes, these cells release their insulin stores. And why this is an important result is during differentiation, you get to a stage where you have immature beta cells, and these cells can produce insulin, but are not readily able to secrete the insulin. So when you get to the point where these cells quickly secrete their insulin, these are cells that are ready for transplant or to help treat patients. If I could direct your attention over here. If you look at panel D, we have a scanning electron microscopy image of the pores of the scaffold. And we seed cells within these pores, and over time they form cell-to-cell connections, as well as these cells form connections with extracellular matrix proteins, the same type of proteins that are found in the islet of Langerhans. So we form a tissue that is fully functional. And here we have a couple videos. So we do confocal microscopy to look at Z-stacked images. So basically we take something that's 3D and we take many pictures. And these videos here can show you what the clusters look like in 3D space. So let me just pull up one more here. Here you're looking at several images from top to bottom. So the green you're seeing is we used an antibody specific for insulin. So any green you see within these clusters are insulin producing cells. And these are within pores of our scaffold. So you can kind of see the shape. Several clusters are coming about. Here are 4 clusters within the scaffold. You can see the entire round shape. And the green here is also positive for insulin. Here we looked at an extracellular matrix protein, which is important for the cell health and the survival. And we found that the cells themselves are producing these proteins. So in a way, the beta cells are creating an environment that's conducive to their long-term success. And this last video will show. We found that the cells tend to produce this protein towards the outside of the cluster, which is similar to what we see with the basement membrane of the islets of Langerhans. So what's really amazing is we put these cells into our scaffold and we provide them with the right media formulations, and they start to mimic what we see in the human body.
I'm taking a tour of the University of Michigan and looking at the incredible research facilities. You know, JDRF funds significant research here. JDRF is all about type 1 diabetes. And Michigan has an extraordinary field of researchers, and we've seen some of those today.
JDRF has been a fantastic resource for the science in the lab. I think I described the 4 areas, and they've all benefited from a very broad network of collaborators, many of whom we identified through the JDRF Encapsulation Consortium. And so we're working with immunologists, with islet biologists, with stem cell biologists, with transplant surgeons. And essentially JDRF has essentially a connection network that we're able to tie into to really meet the right people. And at the same time, they're also really set up well to help you advance the technological discoveries that you have, because they've got connections to get assistance with FDA regulation, with manufacturing, that can allow you to go from essentially a research lab environment where we just essentially visited today and sort of toured sort of these technologies that are in development, and then JDRF has the right infrastructure and connections to be able to sort of take these laboratory discoveries and move them up through the process where they can actually get into patients.
We've seen a whole process, and it's been extraordinary. We've seen the way they're making hydrogels. We've seen tolerizing compounds. We've seen, I would say, the hope for the future, where someone might get an injection of a few cells, and those cells could become insulin producing. And in type 1 diabetes, once you have a diagnosis, then your body no longer really makes insulin. So all of our constituents, our family members, I'm a mother of a daughter with type 1 who was diagnosed at age 10, and after her diagnosis, she is on insulin, exogenous insulin through a pump, and will be until we find a cure. So I think I saw the future today. I'm pretty sure I did. And a lot of bright young scientists and incredible research going on here. I thought we were going to see research that would be years or maybe decades in getting into trials. But you talk about innovative perspectives. These young investigators, this has been totally an eye-opener.
The science we do tells its own story, and I think it's exciting to be able to share with people, here's the progress that we've made. I think it gives me a lot of hope that tomorrow is going to be better than today in terms of what patients are facing and where they can be treated. And at the same time, be able to have this touch with the reality that each of these families is facing and the urgency that they feel. I think it's sort of, the idea is sort of sharing the technologies. I hope it gives them hope, and I think it gives us the reassurance that we're on the right track and essentially tackling the right problems as well.
I was being interviewed when I first assumed my chair of the board role, and someone said, don't you want to see a vaccine so that your granddaughter might not have this disease? And I said, absolutely, I'd love to have, you know, like polio, all kids vaccinated. What I really want, though, is for my granddaughter to see her own mother on the other side of diabetes. I'm here for the cure. And so, you know, what I see today connects me with the work that I do every day, because I'm delivering on a promise. When she was 10, I said, you know, if you can do your part, I can do mine. And at the time, I didn't even know what that meant, but now I do. It's, you know, talking about progress and talking about hope and seeing it tangibly on the bench and the laptop. So it's been a great day.